Thank you, Beppy, and good afternoon, everyone. Yeah, so I will be talking about an International CML Foundation initiative that we've been working on over the last few years, which is a Genomic Alliance. These are my disclosures. Now, we know that genomic profiling using next-generation sequencing has become a standard practice for a number of hematologic cancers, where mutations can define different molecular subclasses, and these can stratify risk and assign therapy. So shown here are the different mutation classes that have been detected in AML. And some of these can be powerful prognostic factors. And just recently, just last month actually, in Blood, an international consensus classification of myeloid neoplasms and acute leukemias was published. And this study not only suggests integration of morphologic and clinical data, but also genomic data. Now, for CML, we know that BCR-ABL is the driving force in the chronic phase. And at this stage, genomic data is not currently used to classify risk for CML. However, evidence has accumulated that mutation of cancer-related genes other than BCR-ABL are associated with treatment failure, and this includes primary resistance. Now, we've just heard from Simona the very important role of BCR-ABL kinase domain mutations in usually acquired resistance. And this is the major known mechanism. But if we look back at next-generation sequencing and what has been performed in CML, one of the first studies was published back in 2011, and this was a study of 39 patients in blast crisis using a targeted panel of specific genes known to have a role in cancer, as well as copy number variation looking for particular deletions. And what this study found was that almost 77% of patients in blast crisis had one or more of these mutated cancer genes. And some of the important findings from this study first were that the frequency of mutated RUNX1 was the same as that of BCR-ABL mutations in blast crisis. Secondly, deletions of a key gene, IKZF1, were very common. Now, these are frequently associated with acute lymphoblastic leukemia and lymphoid blast crisis, CML. And thirdly, BCR-ABL mutations rarely occurred independently of other mutations in cancer-related genes. And we have confirmed this with our recent studies as well. Now, since 2011, there's been a number of NGS studies that have been published over time, and shown here are some of them. So because there were a number of groups that were performing next-generation sequencing and finding mutations in specific cancer genes, we decided it was time to pool our data. So we formed an International CML Genomics Alliance in 2018, and this was facilitated by the CML Foundation and supported by the Patient Advocates Network. And shown here on this map are the groups who first participated in our first meeting back in 2018 that I think was in Stockholm at an EHA meeting. And Professor Baccarani was one of the first people who said that he would attend this meeting. So we first sent out a survey to groups who we knew may have been performing next-generation sequencing. And we actually identified about 700 patients who had had some form of sequencing in genes other than BCR-ABL. So this gave us a hint that there was a lot of perhaps interesting data out there. So the aim of the CML Genomics Alliance was to harness this wealth of genomic information that was being generated in various labs around the world. So what we aimed to do was share and combine the data to improve the statistical power so that we could really understand the clinical relevance of cancer gene mutations. So one of the first things we did was review the literature to determine just what type of data was out there, and we published this paper as an alliance in 2019 in leukemia with the title Laying the Foundation for Genomically Based Risk Assessment in Chronic Myeloid Leukemia. So we summarised the frequency of cancer gene mutations. We identified 15 genomic studies of patients at diagnosis and 20 in studies of patients in the accelerated phase or blast crisis. Now, some of these were using next-generation sequencing, but some of them used even Sanger sequencing of a whole gene. So that was a very brave way to look at mutations in a single gene. So shown here is what we found. So these are the mutations either at diagnosis in red or in blue in the advanced phases, in the specific cancer-related genes in patients who had, where we found at least one mutation in a specific gene. So not unexpectedly, in the most frequent in accelerated phase and blast crisis were the BCR-ABL kinase domain mutations. But at diagnosis, one gene stood out. Now we know that BCR-ABL kinase domain mutations are not found at diagnosis, but 10% of patients had mutated ASXL1. Now this is a gene that is commonly mutated in various hematologic cancers, and it is known to confer a poor prognosis. In the advanced phases, of course, BCR-ABL, as well as ASXL1, but also again RUNX1 and deletions in IKZF1. So collectively, these cancer gene mutations may be more frequent in advanced phases than BCR-ABL kinase domain mutations. But what we were not able to determine really was what was the significance of these mutations at diagnosis, and the reason being that most of these studies were selected patients based on their known outcome. So most of these patients did have a poor outcome, and that was the reason why they were selected for sequencing. But what that meant was the data was really insufficient for evidence-based decisions, because this requires analysis of consecutively treated cohorts rather than highly selected patients. And here's an example. I'm just going to show a couple of examples of some of the studies using highly selected patients at diagnosis using NGS. So shown here is data from our lab where we used whole exome sequencing, copy number variation, and RNA sequencing. And we compared patients who had the very best outcome. Most of these patients achieved a major molecular response by 6 months of first-line imatinib. And then we gathered a group of patients with very poor outcome. Most of these patients had blast crisis, and many of them within the first 3 to 6 months after starting TKI therapy. And so 46 patients in total. In red are shown the mutations that we detected, and we found a significantly higher frequency of mutations in these cancer-related genes at diagnosis in the patients who subsequently progressed. But interestingly, even at diagnosis, we found 2 patients who had ASXL1 mutations. They were treated with first-line imatinib, and they achieved a major molecular response by 3 months. So from our very small data set here, it's really difficult to make clinical decisions based on if a patient has an ASXL1 mutation at diagnosis. So this is where we need a lot more data for evidence-based recommendations. This study was published from the Hammersmith Hospital, and again used highly selected patients, either those who had a very good outcome or a poor outcome. Their cohort of patients, 62, treated with first-line imatinib, for those patients who had mutations in specific epigenetic regulator genes, had a significantly poorer progression-free survival. But interestingly, this difference was not seen for those similar group of patients, 62, who were treated with first-line, second-generation inhibitors. There was no significant difference in disease progression. So this was a really important study. because it does start to suggest that perhaps mutations in key genes at diagnosis could actually influence treatment decisions. And some patients with perhaps high-risk mutations may benefit from second-generation inhibitor treatment in the first-line setting. And in studies of advanced phases using multi-omics, a study from Singapore identified that blast phase is characterized by additional translocations as well as specific gene deletions. And again, these are the same genes that we see deleted in acute leukemia: IKZF1, CDKN2A and B, and PAX5, and others. And by using RNA sequencing, we also identified gene fusions. And again, these are the typical fusion partners that are seen in both acute myeloid and lymphoid leukemia involving KMT2A, which is MLL, again, RUNX1 features, and as well as CBF-beta MYH11, which is commonly seen in AML. And in our study of 39 patients in a blast crisis, we found that 100% of patients had one or more mutated genes. So we found frameshift, nonsense, splice site, and missense mutations in 72% of patients, and there's a relatively small set of cancer gene mutations. Cancer genes that are mutated, and again, RUNX1, ASXL1, IKZF1 deletions predominate. Fusions, we found 31% of all patients had a cancer gene fusion, and 36% of patients had focal gene deletions. Now, these were mostly seen in lymphoid phenotype blast crisis. We also found that 49% of patients had BCR-ABL kinase domain mutations, as we would predict, but every one of those patients had one or more other mutated cancer-related genes. Now, another study published last year was a Japan-Taiwan collaboration, found the prognostic relevance of genetic abnormalities in blast phase. Now, this was one of the largest genomic analyses of blast phase CML. It involved 136 patients, and it did also include previously published data. And they found that genomic abnormalities had a prognostic impact on overall survival. So for patients who had either ASXL1 mutations or mutated TP53, had a significantly worse overall survival than other patients. And again, this was a really important study because it demonstrated the power of combining data sets. So this is what we know so far. There are certain mutations and genomic rearrangements that impact outcome in CML. So these are single nucleotide variants, small insertions, deletions, gene fusions, and specific gene deletions. But there's still a lot that we don't know, and we hope that we will be able to address some of these questions through the Genomics Alliance. So we still really need to determine what the clinical relevance is of mutations at diagnosis on large unselected cohorts. There may be some mutations that confer a poorer prognosis than others that may explain why some of the patients we saw with mutated ASXL1 had very good outcomes. It may be that having more than one mutation confers a poorer risk. And potentially, this poor risk may be overcome with first-line more potent inhibitors. And certainly there may be a role for other drugs that target specific mutated genes. So through the CML Genomics Alliance, we have the potential to answer these key clinical questions by combining data for analysis. So to combine our data, we've partnered with the European-based Harmony Big Data project, and we thank Jan Geisler and Rüdiger Hellmann, Andreas Hockhaus, for letting us know of the Harmony Big Data project that was underway in Europe. Now, at the initial stages, it didn't include CML, but Harmony Plus started a couple of years ago, and that this has now expanded to CML and MPN and a few other diseases as well. Now, how Harmony works is that it aims to accelerate the development of more effective treatments for patients with various blood cancers by combining data sets. And it works because it's a central repository of anonymous data where multiple groups contribute genomic and clinical data in a very secure way. And this follows all of the legal and ethical requirements. The data is quality checked, it's harmonised, and then it's ready for analysis. And importantly, contributors are reimbursed for all of the time it takes to get this data together. And access to the data is secure, and it's only for approved projects. So the ICML Genomics Alliance submitted a project to Harmony Plus, and this was authored by Thomas Ernst from Jena, and it's titled Clonal Hierarchy in CML. Now, it has a number of aims. The first one is to look at these BCR-ABL independent mutations at diagnosis to really establish the clinical relevance of these mutations according to the potency of TKI and how this knowledge could influence therapeutic decisions. We'd also like to look at the evolution of BCR-ABL independent clones during TKI therapy and identify candidate genes that could provide clinical benefit if routinely monitored in patients with emerging resistance, so it could supplement and complement BCR-ABL kinase domain mutation analysis. And of course, we would like to expand looking at molecular aberrations in advanced phase diseases and identify targetable genomic lesions, and potentially even look at single cells and specific cell populations. And this may be relevant for patients in remission when we know that clonal hematopoiesis may develop or even pre-exist. So far we have data submission is underway, and genomic and clinical data of potentially more than 1,000 patients could be submitted. So there are 12 sites who are going to submit their data. This is representing 10 different countries. They're in various stages of submission. We have also identified statisticians and bioinformaticians with expertise in the analysis of CML data, and they're currently learning how to use the Harmony platform for the analysis of data when the submission is complete. So Markus Fuhrmann from Germany and Chung Cook, who is a bioinformatician from Australia, are contributing to this. So we're really aiming to have this data submitted, mapped and harmonised on the platform, ideally by January 2023, so that we can really start the analysis and start answering some of these key clinical questions. But interestingly, if we look at the US-based National Comprehensive Cancer Network, their recommendations already suggest NGS myeloid mutation panel testing for CML. So they suggest that NGS with myeloid mutation panel should be considered to identify BCR-ABL independent resistant mutations in patients with no BCR-ABL mutations. But there are some problems with this. There is no guidance provided for patient management if a cancer gene mutation is detected. And we know that cancer gene mutations frequently co-occur with BCR-ABL mutations, and these could potentially modify response to subsequent TKI. So I think it's important to not only test for these cancer gene mutations in patients where we can't find a BCR-ABL mutation, but perhaps in these patients with mutations as well, because the effect may be additive. A myeloid mutation panel will not detect the deletions seen in patients with lymphoid phenotypes, and we also know that cancer gene fusions are associated with disease progression. So there's still a lot of evidence that we need before we can make these decisions and to aid these clinical guidelines so that these recommendations can be more complete. So there's still an unmet need for patients with cancer gene mutations. And I'd just like to share with you the type of email that I'm now receiving asking about mutations. So I received an email earlier this year from a young European patient who was 6 months from diagnosis still in chronic phase, but had failed first-line, second-generation TKI therapy at 5 months. No BCR-ABL kinase domain mutation was detected, so her clinician ordered a myeloid mutation panel. And this revealed 2 ASXL1 nonsense mutations with a relatively low variant allele frequency of 7%. So the patient asked me, what should they do? Because their clinician has no experience with ASXL1 mutations. What is the recommended drug? And we simply can't answer this question for CML at this stage. The second question, is a stem cell transplant advisable in this case? So I think these are the questions and the management decisions that clinicians will need to consider for patients who have myeloid mutation testing now that the guidelines in the US are actually recommending myeloid mutation panel testing. So we hope that with the CML Genomics Alliance we can combine many data sets, and we thank the Harmony Project for allowing us to participate in their big data project, because we may be able to facilitate the incorporation of genomics-based algorithms into the clinical management of CML based on strong clinical evidence. And I'd like to thank many people who have contributed to this over the last few years. There's too many here to mention. In particular, Nicola Evans, who is from the CML Foundation, coordinating a lot with the contributors, the patient advocates and the Harmony team. Thank you.
Thank you very much, Susan, for this beautiful presentation. Questions? I would say, oh, please, Michael.
Sue, that was fantastic. Just following up on your last points. In blast crisis, I imagine it's difficult to discern anything clinically that might make you suspect additional mutations. But in the chronic phase or the newly diagnosed or early treatment patients, your data, or are you aware of any other groups that have seen clinical changes in blood counts, thinking along the lines of clonal hematopoiesis, first, just a marker versus clonal cytopenias? Is there any way we could predict? Because the guidelines are difficult and broad testing may not be feasible.
Yeah. This is something that we haven't had the opportunity to be able to assess. And it's not actually, we're not actually collecting a lot of that data through the Genomics Alliance database, but it's certainly something that is worth considering, and I'm sure that there will be groups out there that perhaps will be able to look at this. I know the Germany data through the Tiger study has, it's a very large study. They have sequenced 200 patients, and with a controlled clinical trial like that, I suspect that data is available.
Other questions? We know that, I would say, BCR-ABL mutation are causing a genomic instability that can cause, and frequently we have, however, concomitant BCR-ABL mutations and mutations, other mutations, we can say, in the genome. Do you think that, can you give us an estimate of what is coming first and what is probably the cause? Because if there is a mutation of the other genes that can cause BCR-ABL mutation, or is it always the opposite, BCR-ABL mutations which are causing other types of lesions?
Well, yeah, it may not be the mutations themselves, it may be some other mechanism, genomic instability. I guess they all link together, and particularly because we're seeing this strong co-occurrence of BCR-ABL mutations and other mutations. We know that BCR-ABL cells have high levels of reactive oxygen species. We know that can lead to DNA damage. So perhaps it leads to a broad range of damage, not only within BCR-ABL, but in other genes as well.
Thank you. One more question by Michael.
Susan, fantastic. So it's going to take a while to accumulate the data to really predict what to do, or help us make these decisions. But in the meantime, are there mutations that you would already respond to or react to, such as RUNX1 mutations or ICMLf mutations? Would you think if you see them in a chronic phase patients, that would trigger you to strongly consider transplant?
Yeah, I think perhaps an ICMLf deletion. I think that would be very strong. Surprisingly, we have done more sequencing since the data that I just showed, and we have seen a few patients with RUNX1 mutations at diagnosis, and I had predicted that these were the patients who were going to go on to have a poor outcome, but it's not always the case. So, this is where it may be that one mutation may not be enough, maybe something else. We've certainly seen RUNX1 mutations appear late, just at the time of blast crisis. We've seen the same with ASXL1 mutations. We see them strongly acquired at blast crisis, blast phase, but we also see them at diagnosis. So, at this stage, it's really hard to tell. We've seen IDH1 mutations at diagnosis as well. Some of them do extremely well. So it's been really difficult with small cohorts. So this is where we hope that, if we can get 500, 1,000 diagnosis samples, I think we will really be able to nail it.
But say acquired RUNX1 or acquired ICMLf, so absent the diagnosis and then you don't respond very well and you see that for the first time, would that make you think about transplant?
Well, I'm not a clinician, so I can't answer the question about a transplant. Yes. Tim, would you transplant somebody who acquires a RUNX1 mutation?
I think it's a lot more ominous to have something emerge like that rather than something which is, as you say, some of those patients have done very well with the ASXL1 at diagnosis, but we don't have enough data, but I would suspect, I'd be very worried about a patient who we clearly didn't have it, and then it evolved at, say, 6 months in the association with a failure to respond, but a speculation still.
Yeah. And that's where we're hoping to do a lot more samples. At the moment, we've done diagnosis, blast phase, so we need to also test those patients who have emerging resistance.
Edmund.
So great presentation and great initiative. So my question to you is that is this exercise even more important in the emerging economies, because we know for a fact that we are diagnosing our patients late in the chronic phase where possibly the genome is more unstable and mutations are there.
Yes.
And how do we get on board with this?
Yeah, so I predict that you're right. With a more advancing disease, I suspect that we will find more. And, maybe this is something that we need to talk to pharma companies, because next-gen sequencing at the moment is expensive, so we need support. But I think in the long run it could benefit patients. So if we can get some support for these studies.
Thank you.